Hi, everyone. Greetings from Seattle. This is, for those of you who haven't been to our fair city, this is what it looks like on a typical day. I wish, man. Just so you guys know where I'm coming from, I've been at the Fred Hutch for 30 years. Clinically, I'm a marrow transplanter, and then I run two labs. One is an R&D, molecular biology lab, and then the other is a CLIA lab. But it's a little bit different CLIA lab. We actually don't do samples on patients in the clinic. All we do are large clinical trials. So we just do molecular biology for pharma, for the US intergroups, for international studies, and so we just restrict that part. So in the best of all possible worlds, stuff from the R&D lab filters down into that. But we had a lot of chance to use kind of interesting technologies in this role for a long period of time. So when people ask me what I do, I tell them that our lab really studies the genetics of luck, and that is why people who should do well with a particular type of leukemia don't, and why people who should do awfully do well. Try to understand what that is, because that's not what they eat or what they believe in and stuff. There's something driving that. And if you could really put a whole 3 decades of work into one slide, this would be my slide, which is either shows great focus or is really sort of sad and pathetic. I'm hoping the former. So this is what every patient with cancer does, right? You take 3 tracks. You can be completely refractory to chemotherapy. You can do this, which you want, the cure, or you can do what most people with leukemias do, and many types of tumors. They relapse, or they respond and they relapse. And so what we try to do is try to figure out on the front end why people take one of these courses, and these people who have gone down this glide path, why some groups do that and some patients do that, right? So that's what I do. So from a starting point, we've had a very simplistic view of cancer as sort of this giant monolith. And so you go from a stem cell and you start collecting mutations, and then you build this giant identical clone of cells, right? So in leukemias, that's 10 to the 12 cells, 1,000 billion leukemia cells, right? So it's just for point of reference, the Milky Way has 10 to the 11th stars. So there's one order of magnitude more leukemia cells in a patient when they're diagnosed than stars in the galaxy. So that's kind of how we thought. Now, this model works out fine for the extremes. So you can imagine someone who has a homogeneous population of billions of cells, and they're all resistant, and so they don't respond. And you've got the flip side, right? You've got someone who just happens to be lucky and has a homogeneous population of cells that are sensitive, and so you give them chemotherapy, and they do that. Doesn't explain this, why some patients respond and then their disease comes back. That can't be explained by any kind of monolithic clone of cells. People have found now in looking at tumors that in fact they are much more heterogeneity than was early suspected. This is in solid tumors. And just to kind of walk you through this, down here, we can talk about the idea of a founder mutation. We're talking about here's the main prominent clone, and these are all different patients, and yellow doesn't show up. And it says for any particular mutation, 50% or 70% of that cell population has that mutation by bulk sequencing. And then it fixes that population in these individuals to 100% and says, here are different clones you also find. So you can find in some patients, they've got 5 or 6 or 7 different clones at the type of diagnosis, right? And you can do this another way, if I could advance it there. This is looking at a patient who has multiple primary and metastases biops. So they go through and they take multiple pieces from all of these tissues. This is stuff by Charles Swanton. And so each little lane here is a mutation of a particular gene. Each of that swabs there, so these are like 30, 40 mutations. And then if you've read, they have a mutation. If they're green, they have the wild type. And you can see there's different patterns of mutations in the same patient, depending on where you go from the primary to secondary metastases. So there's some selection that goes on with types of different mutations that are occurring in other places and are taking hold. So this is the type of thing you can get from that. You can kind of build a family tree. This is done in I believe, a kidney cancer patient. And you get this sort of branched evolution. So you don't get that one linear tree at all. You actually get multiple clones that are related to each other, but are genetically somewhat different. So you can see this in leukemia, which is my kind of favorite disease. And this is looking at using next generation to kind of track allelic burden and disease burden, and looking at three types of kind of things that can happen in patients. Here's a patient that all these are different mutations, and then this gray is sort of the average of all those. This is a patient who gets chemotherapy, and they completely go away, and they stay away. That's obviously what you want. This is a patient where all the mutations go down and then come back up. So that's probably one population where these differences are probably just differences in the sequencing calls, but it's probably one clone or two clones. And then there's this one who is the sort of relapse shape U curve we showed, where some clones go down and go away, some clones persist. And so this shape is basically made by the disappearance of the sensitive clones and the maintenance of the resistance clones. So in this case, all that's happening is the tumor is using Darwinian selection to its advantage. Because in these cases, the predominant clone at diagnosis isn't the clone that's going to kill you. The clone that's going to kill you is the one it's smaller, and once you eliminate the sensitive clone, it gets more space to grow, it gets more food to eat. So cancer can use Darwinian selection, how we treat patients, to its advantage of survival. So it's using Darwinian selection against us. This just shows that different types of mutations behave differently. These are the proliferative mutations in AML, and they all are pretty sensitive to chemotherapy. But these other kind of founder-initiating mutations, many of them don't do anything once you give them chemotherapy. They are very, very resistant. This is a so-called fish plot of a case with AML, myelodysplasia, who goes on to AML. And again, the different clusters, or there's different color codes of different mutation combinations, showing how some of them rise when they go from MDS to AML. And again, these are just VAF plots of the different mutations over time, showing how the progression from AML to MDS, you get different skewing of the different mutation populations. So you're getting different populations that are essentially competing against each other in the same patient. So this is what you've really got, is many related clones, different sizes, different growth rates. And so you essentially have a cancer ecosystem. And I suspect that if we studied this hard enough, we'd realize that most ecosystems, there are clones that are competing against each other. There are also clones that are probably cooperating with each other. And so curing cancer doesn't mean eliminating all cancer cells, because if you have 10 to the 12th leukemia cells, you probably mathematically can't do that. But the idea is to let clones persist, maybe, that prevent the aggressive clones from flourishing. So if you look at now data on the heterogeneity of the subclones, in AML at least, some have unique phenotypes, immunophenotypes. They have altered properties in vitro. Some of these clones grow faster than others. Some contribute to relapse, some don't. And again, we don't understand the rules of this behavior, and the idea that we really maybe should start, given the clonal complexity, really considering cancer an ecosystem that we have to make an impact on. Now, how would we do? Well, fine. So the problem with all the stuff we've been talking about with next generation sequencing is we're doing it in bulk populations. So we're just basically sequencing the average of all the populations. And so the whole genetic landscape we're inferring from a bunch of assumptions. And some of the assumptions are this. So we take next generation sequencing, we compare the variant allele frequencies, and we try to draw a clonal tree based on the bulk. We're making a lot of assumptions. Probably many of them are wrong. One is, I didn't keep up here, is that we assume that the measurement of the VAF is the same in all genes. You can go to these companies and say, have you looked at your variant allele frequency and done dilution series from 100% to 1% and show me all the ones that are linear and all the ones that aren't? And they haven't. We have for some of them, they aren't linear. So you make major assumptions on a lot of things that have no basis in actual reality of how the test works. Most of these clonal structures are based on essentially assuming that the mutations are heterozygous in all cases. We know that's not true. We know, for instance, that FLT3 ITDs have homozygous with the wild types deleted, right? The VAF is usually in the order of acquisition based on the one that has the highest VAF. That's probably an assumption that may or may not be true. We assume that all mutations are from the same clone. For instance, if you have an NPM mutation, all the calls that have the mutant, you're assuming that's in the same clone. But NPM, 80% of patients have the same four base insertions. So if you had parallel evolution of two different tracks and had the same four base insertion, you would say that those things are happening all in one clone. And we've shown that that's not true. Right? So that's another assumption that just isn't true. Here's an example of how you can get more information from single cells. This is a patient who came in. The FLT3 ITD is a juxtaposition of from 30 to 300 base pairs head to tail in this region. And here's a patient who has actually many patients who actually have 2 ITDs in the same population. So you would say, well, why, if FLT3 ITD has an advantage, why would you ever have a clone that has 2 of them, right? And the answer is, of course, you don't have one clone that has 2 of them, you have more clones. So here's looking at capillary electrophoresis of the bulk, and you see a wild type, 1 ITD and a second ITD. And then if you do brutal brute force sequencing, where you basically do 1,000 to 2,000 single cell sorts and then do genotyping by hand, which is brutal, you find that, in fact, you have all possible different combinations. You have a wild type, you have a heterozygous for the long ITD, a heterozygous for the short ITD, and 2 that have heterozygous without a corresponding wild type allele. And you say, well, that's completely nonsense. So how you can look at that is you can actually count up each cell of each type and average that arithmetically and say, compare that to the bulk. Here's what you get. Here's the bulk read on all these patients. Here's the single cell reads for FLT3 and for NPM1. They're almost identical. So that can't happen if it's just nonsense. So there's way more heterogeneity, even just looking at those 2 genes in this population, than you would ever imagine. Let's go back one thing. One thing that I wanted to point out here that I forgot to is, the other issue is, if you imagine, for instance, FLT3, just one gene, and you have a population, you cell sort, you can have wild type, heterozygous, or homozygous ITD. For any bulk allele frequency of the mutant, you can get that from 3 completely different population constructions based on whether you have wild type, heterozygous, or homozygous mutant. These are 3 different constructions of percentages of those 3 wild types that will give you identical allele frequency. They have to be different biologically, right? But you erase all that once you do bulk. Sorry. So here's another way to look at this. This is a patient looking at 2 different mutations and looking at all the different mutation possibilities. The size is based on this clone size. The color is whether they're hets or homozygous or wild type. And here's sort of a map. And what you can see here is the wire diagram here is doing bulk sequencing and saying, I'm making the assumption this is all one clone. What frequencies of these 2 allele types, FLT3 and NPM1, should I have? And then these are all the actual genotypes, and they are way, way different than you find in the population, with a p-value that most statisticians would believe. So then what we have then is competing populations, right, that come down and go up, come down and go up again. And so what we really want to know is not only what's here, we want to know what's left over after therapy, what's survived. What clones have, and then if we talk about targeted therapy, in something that's this genetically complex, there's too many targets up here. What you want to do is identify the clones that are left over here and then target those, right? Here's an example if you want to look at this type of targeted therapy, and probably the best idea we have is with CML. We have a tyrosine kinase that affects BCR-ABL. When they become resistant, they also escape with a single base pair mutation in the ABL tyrosine kinase domain. Okay, so that's real Darwinian selection. You cannot bind to tyrosine kinase. So here's a patient, these are CML patients who had imatinib and then they relapse with a mutation. You have about half of them have mutations, half of them don't. Then you give them on another second generation drug, and then you find out that in fact you play whack-a-mole and now you have other mutations that arise. This time the 315 mutation that basically dasatinib has no effect on. So as you go with competing tyrosine kinases, you keep selecting out resistant clones. So by the time you become resistant, you have multiple clones, and you're just putting new selective pressure that allows the resistant clones to escape. And this is just, I think, a clean model of what probably goes on in every cancer, is that you have multiple clones, and you're just playing a Darwinian selection game as you treat. So here's different types of trees that have been formed in various diseases. And I compared that to here Darwin's model of finch evolution in his notebook. You can see that, again, Darwinian is right. And one thing I really love about this, Darwin has written in his notebook, I think. I think I have the best idea in 2,000 years. Take that. So in a summary, cancer is made of many related, genetically, biologically distinct clones. Treatment responses probably thought of simple Darwinian selection. We need to figure this out so that we can use Darwinian selection for our advantage, not against us. And to do this, we really have to understand clonal structure and how it changes with therapy. And so how you can do this is you can move away from bulk sequencing, make single-cell analysis routine. The brute force stuff is not sustainable by any postdoc who wants to stay sane. Provide higher resolutions of the clones, and get some technology that we can really do speed this fast, accurately, and at a reasonable cost. And these are some of the questions that we have. One of the issues is, how are we going to measure the clonal diversity? How do you measure a tree shape and what it means? What's a lot of clonal diversity versus a little clonal diversity, right? Are similar trees the same? So here we've got a palm tree, a chestnut tree, and a baobab tree. They look, so there's probably some biological difference between those if you think about clonal tree structure. But even for the same type of tree, does the number of mutations, does the type of mutations that it has make one tree different from the next? Not only that, we found now in myeloproliferative diseases that the order of acquisition of mutations makes a huge difference in the biology. So it may not be just that you have mutants in gene A and B, but what order they occur in makes a difference in how they behave. So the complexity is really, really daunting, but I think reachable. How do genes compete? How do they cooperate? And do we really need to kill all cancer cells to cure? I suspect and hope not, because otherwise we'll never be able to cure them. And I'll leave you now with Dennis to show how it's actually going to get done.